Hello everyone. My name is Petrus. I'm a PhD in Chung Yuan Christain University. I want to show you my study about optimization of laser-based method to conduct skin ablation in zebrafish and development of deep learning-based method for skin wound size measurement. My study will focus on skin wound healing using zebrafish as animal model. This study has been published in Inventions, so you all can read it freely online. Let's start. Skin is known as the body's largest organ in human. It is a vital organ for protection, defense mechanisms, and survival. Skin functions as the fundamental mechanical defense from external factors, such as pathogenic microbe invasion and various environmental hazards. Other than that, skin helps regulate temperature, acts as a sensory organ, prevents loss of moisture, and production of vitamin D. Their function are very crucial to organism. Therefore it is important to sustain their integrity. However, we must have been experience an accident that can cause an injuries to our body. Once the skin got injured, they can lose its integrity and results in functional imbalance. To tackle this problem, our body have a mechanism called wound healing. Following the injury, the skin regenerates immediately to gain protective and barrier function. The wound healing was initiated to prevent further damage and subsequently induce regeneration in the injured area. Because of skin importance function for organism, researcher has been studied the wound healing mechanism extensively. In order to study the step and mechanism of wound healing, rodent has been used by researcher to investigate the process. Rodents share a similar genetic makeup as humans, so researcher often used rodent like mice and rats to do wound healing experiments. Several study in mice has been investigate several factors like temperature and antioxidant. According to the previous result, these factors like temperature and antioxidant plays an important role for wound healing process. In order to induce the skin ablation to mice, researcher often using scalpel, biopsy punch, and dermal laser. Specifically, dermal laser has become the main tools for inducing the skin ablation. However, it is quite difficult and expensive to obtain the dermal laser. Therefore it is intriguing and interesting to find another alternative tools for skin ablation. Furthermore, the usage of mice has some disadvantage due to economic reasons as well as animal welfare issues. Scientific community continuously searches for other alternatives that are able to fit the 3R, which is the replace, reduce and refine for experimental animal usage. Since mice is quite expensive and hard to maintain, it has also become problem to use mice especially for wound healing experiment. In other hand, zebrafish has emerged as an alternative animal model in scientific field. They are relatively easy to maintain and cheaper compared to mice. They have a faster growth. They also have a fully sequenced genome, which make it easy to do a manipulation of its genome. Rapid embryonic development with relatively transparent body during the larval developmental stage, which make it easy to do the observation. These characteristics make zebrafish such a good model for science. In wound healing studies itself, several works has been done using zebrafish as an animal model. Prior study has been successfully introduce a full thickness skin injuries by using dermal laser. According to the study, they found that all steps of mammalian wound repair also exist in zebrafish. This means that zebrafish also possible to used as animal model for wound healing. Furthermore, zebrafish also has been used to examine several compound effect on wound healing like collagen and spirulina. Like I said previously, zebrafish also showed that they have a wound healing process similar with the mammalian. The only differences is the order of the wound healing step. While in mammalian starts with blood coagulation and inflammation, zebrafish start with re-epithelialization and inflammation. But all the wound healing process are actually occur in zebrafish also. This is such a good proof that zebrafish can be used as great animal model for wound healing experiment. Even though the necessity of animal model usage in science is important, but due to concern about animal welfare and increasing cost of laboratory animals such as rodents, another alternative is needed. It is important to have an alternative methods of implementing replace, reduce, and refine in experimentation. Therefore, it is important to establish a new method alternative for wound healing using different animal model and a new tools to induce the skin ablation. Thus, The aim for our study is to develop a new alternative model for skin wound healing by utilizing zebrafish and new rapid and efficient method in skin ablation using laser engraving machine. To achieve our aim, we try to develop and establish the method first. Using laser engraving machine as the substitute of dermal laser to induce the injuries in zebrafish body, between the dorsal fin and anal fin. We then validate our method by testing the known factor in wound healing using our method. Furthermore, after successfully established the method of skin ablation, we also try to develop a method to recognize and measure the wound area automatically by using deep learning or AI. So, here is the video demonstration on how we do the skin ablation on zebrafish using the laser engraving machine. Laser engraving machine we used have a software that can adjust the power, size, shape, and time for the laser ablation. After that we can continue with anesthesize. After the fish was unable to swim we can continue for the skin ablation using the laser engraving machine, and then we just need to observe the wound healing progress in zebrafish after the skin ablation. First thing we do is to find the optimal condition for doing the skin ablation using the laser engraving machine. Here we test whether the laser machine could produce the injuries consistently. According to the result, area produced by laser machine in wood and zebrafish skin were the same. This result showed that laser machine could consistently produce similar size of the area ablated. Furthermore, we also testing the optimal power to use in order to produce consistent result. In this experiment we tested 3 and 5 watt of laser power to find which one is more suitable for the skin ablation on zebrafish. According to the result, both of them could induce the injuries to the zebrafish just fine. However, in the 5 watt group, we can see that the wound closure progress is slower compared to 3 watt groups. Ultimately, there are some significant differences in time needed for the fish to fully recover the skin wound. 3 watt group can successfully close the wound faster. Furthermore, we also tested the histology of the zebrafish tissue after ablation, to check the size of the injuries that successfully induced to zebrafish skin. According to their histology data, it showed that the size of the wound is around 400 micrometer in depth, and 2 mm in width. We can say that the laser machine can successfully produce a full thickness wound to the zebrafish. After find the optimal power condition for laser engraving machine, we do their behavior assessment to evaluate the zebrafish pain response. Behavior assessment was done to examine zebrafish pain response after the skin ablation. We want to make sure that the skin ablation using laser did not affect the zebrafish living condition. This is to meet the concern aboard the 3R of replace, reduce and refine of experimental animal. We want to make sure about their wellness even after skin ablation. From their behavior assessment result, 5 watt group showed alteration in all behavior endpoint tested. Fractal dimension and average speed significantly lowered. This means that after ablation, the zebrafish become less active. Meandering and total traveled in the top also get affected in 5 watt group, which mean they explore the top area less. This alteration may translate it into zebrafish feel uncomfortable after the 5 watt ablation. They may feel pain and changes their normal behavior. However, in the 3 watt group, they did not show any differences in their behavior compared to the control groups. From these results, we can see that 3 watt laser power showed a rather safe outcome compared to 5 watt. Since the 3 watt did not affected the zebrafish behavior at all. Zebrafish after 3 watt skin ablation still swim normally like untreated zebrafish. According to this result, we choose the 3 watt laser power as the optimal power as it did not affecting zebrafish wellness. To measure and calculate the wound area, image J has been used as standard tools in wound healing study. However, since the manual interpretation of the wound eye is easily affected by the opinions of the interpreter, it is better to establish an automated wound area interpretation system to minimize potential biases. Therefore, here we evaluated 2 methods, which is UNet and mask-RCNN. UNet has the characteristic of semantic segmentation, and mask-RCNN has a function of instance segmentation. The result shows that the specificity and sensitivity of the UNet in predicting skin wounds for golden strain are quite high, which is around 0.9 and 0.7. The results demonstrate that the UNet was still able to give a good result in measuring the skin wound size in the TL zebrafish, with around a 0.9 sensitivity. The 0.9 specificities were quite similar to the PET zebrafish. UNet proves to be a good method for measuring wounds on a broad range of zebrafish strains. We put the UNet into test and compare it with image J. Based on 89 images collected from zebrafish by day 5 to 50, it was revealed that there was no significant difference between the ground truth and prediction. Taken together, the optimized UNet performed very well for the skin wound area and morphology prediction. Only significant over or underestimation was found in very small skin wounds. Those errors can be ignored, since they contributed less to the skin wound closure percentage. To put our method into test, we do a validation experiment, examine the effect of temperature to wound healing process. According to the result using our established method, the high temperature showed that it can significantly fasten the wound healing progress compared to control groups. It shows that if we adjust the temperature condition to 35 degrees Celsius, it can help the wound healing. This result was in line with the prior studies that prove high temperature could help the wound healing. Meanwhile, the lower temperature seems to be delaying the wound healing in zebrafish. This result also in line with prior studies that showed low temperature could delay the wound healing, since low temperature will reduce neutrophils, fibroblast, and epithelial cell activities and impair wound healing. According to these results, we confirmed that our method showed a consistent result with the prior studies that already examined the effect of wound healing. Another validation we done to put our method into test. We will using antioxidant to examine their effect on wound healing. Antioxidant known to be effective in help fasten the wound healing. According to our results, both astaxanthin and vitamin C can accelerate the skin wound closure in zebrafish. Days needed for the astaxanthin and vitamin C groups was faster than the untreated group. This result was indeed proof that our method indeed worked consistently with prior studies in wound healing. From this study, we proved that we successfully utilized zebrafish as animal model for wound healing, and we also developed the new alternative method using laser engraving machine. We also successfully develop a new measurement method using UNet, and it shows a good sensitivity and specificity. Validation using temperature and antioxidant showed consistent and similar result with prior study in wound healing using zebrafish and mammals. It proved that our method exhibit consistent result. These are related studies that successfully done by using our laser ablation system. We welcome you for your collaboration with us. It is such a pleasure for us to do some collaborative studies with others. Thank you everyone for listening to this presentation. Cheers everyone.